Hello students, welcome to LEELA'S tutorial. So today we will discuss about regeneration of plants. Normally plants are regenerated in plant tissue culture lab either by organogenesis or embryogenesis. So today we will discuss organogenesis in detail. Right? So first, what are the factors affecting organogenesis? So here the first one is the type of explant you are taking. So already we have discussed explant is defined as any part of the plant body which can be used in plant tissue culture in order to produce a complete plantlet. Right? So why that complete plantlet is produced from a explant? What is the principle involved? That is the totipotency effect. Totipotency, it is the ability of the single cell to get transformed into a complete plantlet. Right? So based on that, here you're going to select the explant. I said any part of the plant body. So you can take leaves, nodes, meristem, roots, right? Any part you can select, and then the next one is the type of media and the composition of the media. So whatever the explant you are selecting, the media must be suitable for that explant. Okay, so it must provide all the nutrients required for that explant to grow or get regenerated into a complete plantlet. Then it is plant growth regulators. So here we have plant growth hormones, positive and negative growth hormones, but here we require only the positive growth hormones, that is your auxin and gibberellins, right? Auxin and cytokinins, where gibberellins are required only for the elongation process, right? So here we don't want elongation, we want shoot initiation and root initiation. So for that we require only auxins and cytokinins, but not gibberellins, ethylene and abscisic acid. Okay, so here the plant growth regulators are also must be monitored in such a way that whenever they are required, only in that time you are going to provide them. Okay, so that you can be doing by transferring the plantlets or the callus, whatever you are growing, that can be transferred from one media to the another media, that is referred as subculturing process, right? Then last one is the genotype of the plant. Okay, so based on all these factors, when you correlate and maintain them, okay, you must maintain them in such a way that the explant, whatever you are going to culture, it must have the favorable conditions, right? So once the complete process is set up, you are performing this procedure. So let us see how you are going to produce a plantlet by means of organogenesis. So here, based on the formation of the plantlet, you have two different types of organogenesis. The first one is direct organogenesis and the second one is indirect organogenesis. In case of direct organogenesis, here if you are going to take leaf as a explant, so only this portion you will be taking as a explant, right? When you inoculate this explant in a media, right? So this explant will have the totipotency effect, right? Totipotency is the ability of the cell to develop into a complete organism or the complete plant organs, right? So here it will result in formation of a plantlet, the shoot system and the root system. Whereas in case of indirect organogenesis, again you are going to take the explant, you are going to excise the required portion of the explant. Then what you are going to do, you are going to induce the formation of the callus. The callus will be formed, then from the callus the micropropagation occurs and it will result, the callus will result in formation of shoot system, right? Then after that the root system. So here the intermediate process, or this is the step which is involved. So it is an indirect organogenesis. Whereas in case of direct organogenesis, there is no formation of callus. So here you can say that leaf is an differentiated tissue. That means it is having structure and also the function, right? Whereas here the differentiated tissue is getting transformed into undifferentiated tissue or mass of cells. That means here callus doesn't have any structure or function, right? So again, from the differentiated tissue or the explant, the differentiated plants are formed in case of direct organogenesis, whereas in case of indirect organogenesis, the differentiated tissue is getting transformed into undifferentiated mass of cells and then again attaining the differentiation, redifferentiation. That means again it is attaining the structure and function. Is that clear? So that is the difference between your direct organogenesis and indirect organogenesis. So let us see in the lab what we are going to perform. So first you are going to select the mature plant. So here the mature plant is rooted in the soil, which is having flowering and fruiting ability without any diseases, right? So now you are going to select the mature plant. You know the genotype of the plant. Then here you are going to select what explant you are going to take. It can be the root, root tip, nodes, right? Axillary meristem or apical meristem or leaf, right? Any part. So here I have selected leaf, right? So from the leaf, the required portion of the explant is excised, right? With the help of scalpel, I am going to cut this, right? So here I take scalpel and excise this one. Okay, when I cut the required portion, I inoculate on the plant tissue culture media. So here we have discussed that the media type and composition is very much important, right? So based on the explant you are taking, you are going to select either the MS media or that is Murashik and Scoop media or White's media or EM media, that is Eagle's minimal essential media, right? And some other supplements can be provided like coconut water, coconut milk, tomato extracts, right? So based on the explant and its requirements, you are going to prepare the media. After standardizing the media, you are going to inoculate the explant. So after inoculation, it is the incubation. So here the main part which is played is auxins. The role of auxins is very much high, right? So auxins mainly induce. So I said auxins and cytokinins. Auxins and cytokinins are the growth hormones which are required. When you are providing auxin and cytokinin in same ratio, 1:1 ratio, then that will induce the formation of callus. The differentiated explant will get transformed into undifferentiated mass of cells. A ball of cells, or you can say a clump of cells are formed, right? So from this undifferentiated mass of cells, when they are formed, right? So this will take minimum of 3 weeks, right? By 3 weeks you can observe the callus formation. Okay, so after the callus formation, from the callus, the meristemoids will be produced. Meristemoids are the green dotted structures over the callus. From each green dot meristemoid will get transformed into a single plantlet, right? So after this, right, after the callus formation, further incubation, right, it will result in formation of shoot system, right? So from each meristemoid there will be formation of shoot. That is nothing but your micro propagation process that we will discuss in detail, right? So by micro propagation, here each meristemoid will get transformed into a plantlet or the shoot system. So now this is called as the shoot formation stage, right? So here as it is present in shoot initiating media, or you can say shooting media, right? Then again the plantlets which are aroused on the callus are transferred into another media. This transferring of plantlets or the callus from one media to the other media is referred as subculturing process, right? So everywhere we are using semi-solid media only. It is not the suspension culture, right? It is semi-solid culture. So here again we are transferring each plantlet individually in separate media. Okay, so again here you are taking semi-solid media, but it is rooting media which initiates root formation. So what is difference between your shooting media and the rooting media? Here you can see at the callus stage, the auxin to cytokinin is same 1:1 ratio. When you are transferring this callus with meristemoids over the shooting media, the media consists of auxin to cytokinin 1:3. Right? So the cytokinins induce shoot formation. After the shoots are formed, the individual shoots are transferred into the rooting media. Again, the rooting media consists of all the nutrients, but here the auxin to cytokinin will be differing in all the stages. So here it is auxin to cytokinin, that is 3:1, where you have 3% or 3 times more than the cytokinins. So here the auxins will induce the formation of root. Okay, so here the auxins play very important role, right? When they are present in different types of media, they result in either the shoot initiation or the root initiation. So once it is formed, now the plantlets which are formed will be shifted to the greenhouse. Okay, so that again acclimatization will study in detail. Okay, so that is they are shifted to the greenhouse, then from greenhouse they will be shifted to the pots. Okay, from the pots they are given to the farmers so that they can grow in fields. Right, so here this is the process of indirect organogenesis where callus is involved. Right, so if it is a direct organogenesis, see here, after the explant is inoculated, you are not going to take the media which consists of auxins and cytokinins. Right, so here when the auxin and cytokinin is 1:1, it will result in formation of callus. Right, so here directly the explant I am going to take which consists of 1:3 auxin to cytokinin. So here this step is omitted. Okay, so when I use same concentrations of auxin and cytokinin, it will result in callus. When I don't use that, then it will not enter into this callus stage. Okay, the differentiated cells remain differentiated only. There is no callus. Right, so from this again the shoots are formed in the shooting media where the media consists of auxin to cytokinin 1:3. Then next again they are transferred to the rooting media, auxin to cytokinin 3:1, right? Then you get the plantlet. Everything is same. Only the thing is, in both the cases, the complete organs. What do you mean by organogenesis? Organogenesis is defined as formation of organs, that is plant organs, that is root system and the shoot system. So here the complete plantlet is produced either by direct organogenesis or by indirect organogenesis. Is that clear? If you have any doubts, put it in comments. Okay, next class we will discuss about embryogenesis.